So welcome. In this session, we'll be looking at DNA replication in prokaryotes. There are some slight differences between DNA replication in prokaryotes and eukaryotes, but some steps are similar, okay, or somewhat related. But by the end of this session, you should know how DNA replication, the steps in DNA replication, which include DNA initiation, elongation, and termination. Okay, so welcome all. My name is Wallace Munyeri, and I'm the Daktari. Now, maybe I'd drawn this. Now, when the two strands of double-stranded DNA are separated, each can serve as a template for replication. Okay, so we must first separate this double-stranded DNA. Then each of the single strand will serve as a template for replication of a new complementary strand. This is what I was trying to do here. In red here are the parent strands. Then they will first separate. Okay, they will first separate. Then they will give they will be used as the template for synthesis of the new complementary strands shown in green here. So the new double-stranded DNA will have one old strand and one new strand. So in this way, DNA replication is said to be semi-conservative. Okay. Then the synthesis, the enzymes which are required for this DNA replication are usually template guided, and they require magnesium. Okay. And they usually synthesize the complementary sequence of each strand with high fidelity, extraordinary fidelity. It is rare for them to make mistakes. Okay. Now, for prokaryotes, E. coli is the one which has been studied, and it is the one which has been used for description of DNA replication in prokaryotes. But DNA synthesis in higher organisms, such as eukaryotes, and it's more complex, but involves the same types of mechanisms. Okay. In either case, either prokaryotic or eukaryotic, DNA replication commits the cell to continue the process until the entire genome has been replicated. So you must have initiation. Initiation of DNA replication is like the commitment step for entire genome replication. Now, initiation. We first must separate these complementary strand. We must first separate the double strand. Okay. This is done. First, we must identify the origin of replication. Okay. This cannot start anywhere. Okay. DNA replication begins at a single unique nucleotide sequence, a site called the origin of replication, or ORI. If you hear me saying ORI, it is the origin of replication. For the case of E. coli, it is ORIC. Okay. And within the ORI, you will have a specific nucleotide sequence. And in animals or in organisms which have a multiple ORI, such as eukaryotes, or those with the linear DNA, that specific nucleotide sequence will be repeated at every ORI. That is why it is known as a consensus sequence. And that nucleotide sequence, it is rich in adenine and thymine, AT rich. Okay. Why AT? Because remember, the hydrogen bonds between AT are 2, but between G and C is 3. So it is easier for us to break 2 hydrogen bonds than 3 of them. Okay. So as to facilitate melting, that is why it is AT rich. Okay. Now, replication fork formation. Now, as the 2 strands unwind and separate, synthesis occurs at 2 replication forks that move away from the origin of from the ORI. Okay, in bidirectional. Okay, this generates a replication bubble. This is what I'm trying to show you here. I think this is one DNA strand and its complementary one. Then along this line, that is the ORI. Okay, so after a local unwinding due to binding of some proteins such as DNA A, the replication fork is formed here. replication fork is formed here, and it will grow towards this side, and the other one, the replication will go to this side, okay? And by doing so, a replication bubble is formed, okay? Now, which proteins are required during this strand separation and replication fork formation? You require one very important protein known as DNA A protein, which initiates the replication by binding to a specific nucleotide sequence within the ORI. For the case of E. coli or IC, that specific nucleotide sequence is known as the DNA box, okay? So DNA box will cause a melting of the, once DNA A binds to the DNA A box, it will cause melting of the AT-rich region within the ORI, hence local unwinding. Then DNA A helicase, this usually binds to the single strand DNA near the replication fork, then it will move towards the neighboring direction, which has the double stranded towards the region which has double strands, okay, the double stranded region, okay. Then it will cleave those hydrogen bonds between the complementary bases, okay. By doing so, it will unwind the double helix. And the helicase, they require energy in form of ATP, okay. So they derive their energy from ATP hydrolysis. Now, the unwinding at the replication fork by this helicase will cause supercoils in other regions of the DNA molecule. And you will see how we deal with these supercoils. Supercoils are of two types: positive supercoils, or overwinding, and the negative supercoils, or underwinding. And these positive supercoils can prevent the effective unwinding of the DNA. So you must remove them, okay? Now, in E. coli, DNA B is the principal helicase, okay? And DNA B is a hexameric protein, okay? And it is usually loaded to the DNA, the double-stranded DNA, using a DNA C, okay? Now here we are, you can see this is your helicase, okay? Or your DNA B in E. coli. So it usually cleave these bonds here between the bases, hence effecting unwinding, okay? And this is the direction of movement of the helicase and also the direction of movement of the replication fork, okay? Then you have other proteins required in the replication fork formation. These single-stranded DNA binding proteins, okay? They usually bind to the single strands and prevent them from reannealing, from annealing to each other, from joining back, okay? So it's not like the helicase is doing no work, okay? So you must first, after separating them, you must hold them in that position to prevent them like from snapping back. And this, apart from doing that, from preventing the single strand templates from reannealing, they usually also protect these single strands from degradation of by nucleases, okay? Now, how do we solve that problem of the supercoils due to this DNA helicase? We have enzymes known as DNA topoisomerases. These DNA topoisomerases, they are of two types, 1 and 2. DNA topoisomerase 1, it usually break, or it creates a transient nick on only in one DNA strand. Then the intact one, the other DNA strands which was intact, has to pass through that nick, okay, so as to relieve the supercoils. Then after the intact DNA strand has passed through that nick, we reseal the nick, and it is the same enzyme which reseals the nick, okay? So by creating a nick, it acts as a nucleus, and by resealing the nick, it acts as a like this, okay? DNA topoisomerase 2 now cleaves. It creates transit nicks or breaks in both DNA strands, okay? And we have a bacterial DNA topoisomerase 2 known as the DNA gyrase. And DNA gyrase is usually a target for some antibacterial drugs such as fluoroquinolones. Okay, such drugs include ciprofloxacin. Ciprofloxacin targets DNA topoisomerase 2 in bacteria, which is better known as DNA gyrase. Now, which is the direction of DNA replication? Now, these DNA polymerase, they usually are responsible for copying the DNA templates and are able, they will only read, please note this, let me take a pen so that I don't confuse some of you. They will read the parental nucleotide sequence in a 3' to 5' direction. That is the way they read. But they will synthesize a new DNA strand only in the 5' to 3'. Okay. Now, therefore, beginning with one parental double helix, two newly synthesized stretches of nucleotide chains must grow in the opposite direction. Okay. So if you have, let's say, yeah. So this is where we start. And let's say a replication fork. Let me draw just one replication fork. So let's say this is 3, and this is 5', and this is 5', and this is 3'. Okay. Now, one strand will be synthesized in this direction, and the other one will be synthesized in this direction. Okay. So the one which is synthesized in the direction towards the replication fork is known as the leading strand. Okay. And the other one which is synthesized in the direction opposite from the, or a direction away from the replication fork is known as the lagging strand. Okay. So leading strand is the one which is copied in the direction of the advancing replication fork, and it is synthesized continuously. Okay, please note this. It is synthesized continuously, and that is why it's called the leading strand. Then the lagging strand, it is copied in a direction away from the replication fork, and it is usually synthesized discontinuously. So we will synthesize it as small fragments of DNA. Okay, usually synthesize it in form of small fragments of DNA, and which are copied near the replication fork. Okay, so this synthesis here is continuous and the other one is discontinuous. So that is why DNA replication is said to be semi-discontinuous, because it is half continuous and half discontinuous. Okay, now those short stretches of discontinuous DNA along the lagging strand are known as the Okazaki fragments. Then they are eventually ligated together by a ligase to form one continuous strand. Okay, so the new strand of DNA produced by this mechanism is termed as the lagging strand. Okay, now here, yes, here is the diagram. You can see this is the origin of replication, and this is 3' to 5'. So this has been synthesized from 5' to 3' direction. Okay, and it is a leading strand. Okay, but this one here, it has been synthesized also from the in form of 5', also can only synthesize in the 5' to 3' direction, but it will be done so discontinuously. Okay, so you will have several Okazaki fragments which will eventually be ligated together. Okay, now DNA polymerase cannot initiate synthesis of a complementary strand of DNA on a totally single template. It requires a primer, an RNA primer. Remember another enzyme which required a primer was glycogen synthase, can you remember? And the primer was being created by the glycogenin, which had a tyrosine at position 194. Can you remember that? Please go and look at that. So the enzyme primase, we have an enzyme known as primase, which is a DNA-dependent RNA polymerase. Okay, usually synthesize just a short piece of RNA-based pairs. It is like about 10 ribonucleotides, and to that RNA primer is the DNA polymerase will add nucleotides to that DNA, that RNA primer. Here you can see the RNA primer, and you can tell it is RNA because you can see these are ribonucleotides, okay? Then for DNA polymerase to work, it requires this primer, and it will add a deoxy ribonucleotide to this position, okay, to form the 3' to 5' for phosphodiester linkage. So it requires a primer. Please remember that. Okay, and that primer is synthesized by a primase, okay, which is a DNA-dependent RNA polymerase. Now, let's look now at the chain elongation now. The DNA polymerase usually elongate a new DNA strand by adding deoxyribonucleotides one at a time to the 3' end of the growing chain, okay? Now, let's look at DNA polymerase 3, which is the main one. Now, DNA chain elongation is catalyzed by a multi-subunit enzyme known as DNA polymerase 3. Using the 3-hydroxyl group of the RNA primer as the acceptor of the first deoxyribonucleotide, DNA polymerase 3 begins to add nucleotides along the single-stranded template that this adding of the deoxyribonucleotide, the sequence will be specified by the parent template strand, okay? So by doing so, it has a 5' to 3' polymerase activity, okay? And this enzyme, it is a highly processive enzyme. That is, it usually remains bound to the template strand as it moves along and does not diffuse away and then rebind before adding a new nucleotide. So it will remain bound to the template strand as it moves along, not that we first add a nucleotide, then it moves away, then comes back. No, it will remain bound to the template strand. Now, the processivity of this DNA polymerase 3 is as a result of the beta subunit of the holoenzyme forms a ring that usually encircles and moves along the template strand of DNA, hence serves as a sliding DNA clamp. So please note that in prokaryotes, the processivity of the DNA polymerase 3 is due to the beta subunit of the enzyme. Unlike in eukaryotes, where you will see a protein known as the PCNP, okay, the proliferating cell nuclear antigen, okay, proliferating, maybe I said wrongly at the first time, proliferating cell nuclear antigen, PCNA, and it will require another protein so that it can be loaded. Now, DNTPs, deoxynucleoside triphosphates are the substrate molecule for this enzyme, and the DCTP, DATP, DGTP, and the DTTP or the TTP. Now, whenever we add maybe I go back to show you something. Here, you can see this is the a DNTP. But whenever we add it, we release a pyrophosphate. And the pyrophosphate is hydrolyzed, we add water, and a reaction catalyzed by the pyrophosphatase, you get to inorganic phosphate. And this energy, there is energy released in this reaction. So essentially, this reaction is irreversible, okay? The reaction is irreversible. So what we add to the RNA primer are deoxynucleoside monophosphates, okay? Monophosphates. We added deoxynucleoside monophosphate. We add deoxynucleoside monophosphates to the growing chain through a 3' to 5' phosphodiester bond. Then proofreading so that we can have fidelity. Now, DNA polymerase 3 has a proofreading activity. the 3' to 5' exonuclease activity. Now, in addition to the polymerase activity, the 5' to 3'. Now, as each nucleotide is added to the chain, DNA polymerase 3 usually check to make certain that the base of the newly added nucleotide is complement to that of the template strand. Okay, if it is not, the 3' to 5' exonuclease activity will remove the error in the direction opposite to the polymerization. So if polymerization is occurring in that direction, proofreading will occur in this direction. So every time we add the polymerase, somehow slides back to check if that was in fact the correct base. Now, let's say you make an error. Instead of now, for example, if the template C and we need a template C and the enzyme say inserts A for G. So here we have a G. So you know you are supposed to put a C here, but you put an A. Put an A. The enzyme will slide back. Okay. Cleave this through the 3' to 5' exonuclease activity. Then move again forward through the 5', 3' polymerase activity and add now the correct one. Add now a C. Okay. That is how it can introduce and eliminate errors from the chain. And these domains, the 5' 3' polymerase and 3' 5' exonuclease are located on different subunits of the enzyme. Now, RNA primer excision and replacement by DNA. After forming these primers and after elongation, you need to remove them. And they are usually removed by DNA polymerase 1, okay, which has a 5' to 3' exonuclease activity. Maybe I should go to the diagram directly to show you that. Here we are. You can see this is a primer. So after this strand has grown due to DNA polymerase 3, the DNA polymerase 1 usually insert there. Then it will cleave this, remove this, okay? And then as it removes this through the 5' 3' exonuclease, it will add them here. It will add them back to the chain that was behind it, as shown here. You can see, add here through the also the 5' 3' polymerase activity, then proofread via the 3' 5' exonuclease. So it is a very busy enzyme. It has 3 activities as shown here. So it has both 5' 3' exonuclease for removal of the RNA ribonucleotides, has a 5' 3' polymerase for addition of the DNA nucleotides, and 3' 5' exonuclease for proofreading. Then after that is done, we need to ligate here. To ligate. So DNA ligase comes in, as shown here. DNA ligase will ligate this, and this reaction requires an ATP. So you can see that is how we form the complementary strands. So just maybe this overview will help you. Look at this, know the function of each SSB, know the function of the topoisomerase here, know the function of the primase. And remember, primase is a DNA-dependent RNA polymerase. This is the leading strand. You can see the leading strands require only one DNA primer, unlike the lagging strands where we have different primers, multiple of them. And then these are the Okazaki fragments. Then remember, DNA polymerase 1 will come here when this DNA polymerase 3 synthesizes the chain and comes close to the primer. It will be inserted there. Then it has 3 activities: the 5' 3' exonuclease, so as to remove the RNA primer. Then also 5' 3' polymerase, so as to add the complementary bases to this side. Then it will also have a proofreading activity, the 3 prime. So it will go back to see if it has added them correctly, the 3 prime, 5 prime exon nuclease activity. So I think termination in the E. coli, it is usually mediated by a sequence-specific biting of a protein known as the TAS protein, the terminus utilization substance, which usually binds to the replication termination site, the TAS site on the DNA, which will stop the movement of the replication fork. Okay, and that is how it is terminated. Also, and because it is circular, because we started somewhere here, the replication fork are growing in this direction, and the other one is growing in this direction. If these replication forks meet here, that will also terminate the DNA replication. Okay, so I think we have come to the end, and ask yourself this question: did you understand that process? Put in the comments, and thank you. And please subscribe for more anatomy and biochemistry content. Thank you.